All right.
So welcome everyone again. Today's speaker is Nathalie Feiner from Lund University in Sweden. She's a evolutionary biologist with strong interest in developmental evolutionary biology. She did her PhD in the University of Konstanz, finished in 2013 in the natural history of vertebrate genomes. Then she moved on as a postdoctoral Humboldt postdoctoral fellow to the Oxford University, where she started to work on Anolis lizards. And she moved her focus a bit to more organismal questions. Since 2015, she's working at the Lund University in Sweden. And in 2021, she started her own research group, funded by very prestigious grants from the Swedish Research Council and the European Research Council. She's using lizards as a model organism, where she studies various aspects of developmental plasticity, evolutionary theory, and also transposable elements and genome evolution. So she has also a recent interest in understanding how development affects adaptation and diversification. And yeah, with that, the stage is yours, Nathalie. Thanks for coming to us today. Look forward to listening to your talk.
Great. Okay. Thank you very much for having me here. And thank you, Ike, for the very nice introduction. So I'm really grateful and glad that Madeleine and Ike invited me to this MEEGene seminar series here. So I'll talk today about a project that we've done on Mediterranean wall lizards. So I think we can all agree that one of the major questions in evolutionary biology is to understand and explain adaptation. And adaptation is fundamentally a multivariate problem. I think we can appreciate this when we look into these seahorses here, if you manage to spot them. So there's two seahorses here. So they are only really fit and manage to blend into the habitat when they have the right combination of behavior, morphology, and coloration. So different traits need to fit together to make a fit organism. So in other words, adaptation requires phenotypic integration. And this is not only the case for crypsis, so that's not a special case, but it's generally true for all kinds of adaptation. And from an evolutionary perspective, we can ask a number of different questions on phenotypic integration. So for example, we can be interested in the evolutionary origin of traits integration. We can ask how it's maintained, evolutionary timescales, and we can also ask how it can be modified and be changed. And when you think about the causes of phenotypic integration, what probably first comes to mind is correlational selection. I'll explain this here by using these garter snakes from the United States as an example. So these snakes, they come in different color patterns. So they can be either entirely striped on their back, or they can have this like more disrupted blotched pattern. And they also show variation in their escape behavior. So when they are startled by a predator, they can either just retreat in a direct straight line, or they can show a number of reversals by just snaking basically away. And what Brody has shown in this seminal work here is that only the snakes that have the right combination of dorsal pattern and escape behavior, only they have high fitness and therefore high survival. So this is really correlational selection in action. On the other hand, traits can be integrated not due to selection, but they can be integrated already when they appear in development for the first time. And this is what we call covariabilities. The clearest example of covariability is when traits are inherited together as one package. And that's the case when we talk about supergenes, for example. There's this famous case of the ruffs, which I know that many of you are familiar with. In this case, there's a number of different loci that are causally important for different traits. And these loci are co-inherited as one supergene because there's an inversion that fixes them together. So phenotypic integration is due to inheritance of one supergene that combines all these traits together. But there's also a little bit more sort of softer cases of covariabilities. And these we can describe as pleiotropies. So the classic case of pleiotropy is genetic, at the genetic level. And for example, a transcription factor regulates not just one target, but many different target genes. But it can also be seen at different levels of biological organization. So for example, at the physiological level, when hormones affect a number of different processes, or also at the developmental level, when, for example, a single or a number of cell types gives rise to many different downstream organs. So I present them here as basically alternatives, correlational selection and covariability. But in reality, of course, they are just two sides of the same coin, because covariabilities are produced by the developmental system and they shape the variation that selection can see. And then over time, correlational selection can lead to certain kinds of covariabilities. So I think it will be important to study both sides of these processes in order to really understand where phenotypic integration comes from and how it's shaped by different kinds of processes. So now I've just given you a little bit of a background on how I like to think about phenotypic integration. And for the rest of the talk, I will present to you a case study that we have done, where we looked at the origin and the introgressive spread of a novel and complex phenotype in wall lizards. So I first start by introducing you to our study system. So we are dealing with one genus here, which is called Podarcis. And those wall lizards are occurring in Central and Southern Europe with a quite broad distribution. Overall, there's something like 27 species currently described. The numbers are slightly increasing with time. And I'm sure many of you have seen them when you visit places like the Mediterranean beaches, then you come across these lizards very frequently. So I'm sure you have seen different species of them. Today, specifically, what I'm talking about is on one species which has the broadest distribution. So this species is called the common wall lizard, Podarcis muralis. And you find it basically everywhere from northern Spain in the west to Turkey in the east, and has a very broad distribution stretching almost up to Bonn in Germany. I guess you will find it outside your door, basically. And throughout most of its distribution, these lizards look rather boring. So even to my eyes, they're not very exciting. They're just brown lizards, relatively slender, with not many interesting features. But if you travel to central Italy, you find lizards of the exact same species that look drastically different. How are they different? So if you compare them, then most conspicuous to your eyes is, of course, the coloration. So these lizards in central Italy, they have a green dorsal coloration. They have UV blue spots on the side here, and they have black ventral coloration. They differ also in morphology. Males reach up to twice the size of these brown ones here. And they have also massively big and large heads and skulls. And where they co-occur, they also show differences in dominance. So these lizards are dominant over the brown lizards, and they show much higher levels of aggression. So they differ systematically in coloration, morphology, and behavior. And they are so different that this has led people to describe these ones here in central Italy as a different subspecies of Podarcis muralis. And so in the talk, as a shorthand, I will refer to these ones here as the green lizards and the other ones here as the brown lizards. But remember that it's a whole suite of traits that discriminates these two kinds of lizards. And the obvious question that we want to address now is, of course, how this brown ancestral type has turned into this green derived nigriventris type here. And although I've now pretended that we are dealing with two different morphs, basically. That is not the case because they are continuously varying between green and brown. And I make this point here by showing you this continuous variation on a map. So here on this map of central and northern Italy, you see that each dot is a single population that we have sampled. And for each population here, we have sampled the dorsal coloration of up to 30 lizards, and we have scored them in a greenness score. So we have checked this with spectrophotometry, but we can also just by eye say that 10 is a very, very green lizard and 1 is a purely brown lizard. And then we can line them up on a continuous base, on a scale from 1 to 10 here. And if we then smooth this greenness score across the landscape, then you see that there's really a continuous variation, with some populations being entirely consisting of purely green individuals. Then we have a gradient over the mountains here. And then in other parts of Italy, we have purely brown populations, but also a mix of individuals in between. So the phenotypes, they are continuous between green and brown, and they vary across the landscape. And remember here again, I show you the green dorsal coloration or the green and brown continuous variation as one trait, but I could show you any of these other traits and you would basically get the same picture as well. And to some extent, at least, we know that this geographic distribution is explained by climate, because we know that the higher the Apennine Mountains here, the more brown are the populations of lizards that you find. So go back again. So the across Rome and here down in this area, we have very green lizards, but then the higher up in the mountains you go, the more likely you are to find purely brown lizards. So climate seems to modulate the distribution of lizards. So what is actually quite surprising is that we are not just dealing with a single lineage of lizards here, but we have, when we consider the phylogeography, we understand that these are two different very old lineages that are getting into contact in Italy. So here I show you a phylogeography of the entire species. And we find that there's 6 deep genetic lineages that are dividing up the species. And we have dated that the oldest split within the species is 6 million years old. So it's a really old split. And this split is between one lineage that is the Southern Alps lineage that is located here in northern Italy. And the other lineage that is meeting exactly here is the Central Italian lineage here. So here we have 2 lineages that are in secondary contact and a hybrid zone in between. And I show you this again here on a little bit of a more zoomed in map so that you can appreciate where exactly the phenotypes are in relation to the phylogeography. So here we have the Central Italian lineage. Then we have a hybrid zone, a relatively narrow hybrid zone. And on top here, there's this Southern Alps lineage that is 6 million years divergent. But nevertheless, we have these green phenotypes that are introgressing into this distantly related Southern Alps lineage here along the coast of Liguria. And now with this as a background, now we can ask about the trait integration of our syndrome trait, because so far I've just stated that we are dealing with a suite of 5 different traits, but now I can show you how statistically significant this actually is. So if we ask for the pure Italian lineage now, how strong the pairwise correlation between different morphological and coloration traits is, then we find that all of these pairwise correlations are highly significant, meaning that a lizard that is more green is also more black and has more blue spots on the side, has a bigger head, and is also larger in size. So all these traits are really going together. And this is not only the case in the Italian lineage, where this nigriventris phenotype is mostly distributed, but it is also the case in the hybrid zone and even in population in the Southern Alps lineage, where these phenotypes have introgressed into. So even there, these pairwise integration of these syndrome-related traits are statistically significant, except for 1 or 2 cases where they are not as strongly pairwise integrated as in the Italian lineage. But we're still concluding here that we deal with a syndrome and that the syndrome remains intact even when it's introgressing into a distantly related lineage. I've mostly spoken about males so far. So what about females? So females show the same kind of trait introgression, but always a little bit weaker than males. So females never get as shiny green as males do, but they also show the same expression of traits. And here I show you on a phenotypic trajectory analysis that basically follows the phenotype change along a trajectory that goes from pure Italian over the hybrid zone into pure Southern Alps. And here I show you how the phenotypes change in morphospace. So here I just plot principal component 1 and 2 for all these syndrome traits. And here you can see that for the different sexes, so here I plot females and here are the males, we find the same change in morphospace, namely that we go from pure Italian here over to pure Southern Alps, where the phenotypes change in the same direction for both males and females, and also in the same magnitude. So females are really showing the same changes as males, and they're not just following the males, but they're really showing in parallel the same changes. So we conclude here that it's really both sexes that show the same pattern of introgression into the Southern Alps lineage. So the next question we addressed then is to ask about the origin of these syndrome phenotypes. So as you can already appreciate from the map, the strongest expression of the phenotypes we find around today's Rome here. So what we did to test if indeed Rome is the origin of these exaggerated phenotypes, we used the following design. So we selected 6 populations, 2 populations in the Southern Alps lineage, and then 4 populations in this Central Italian lineage. And we always picked contrasts between purely green populations, so populations with purely green lizards, and purely brown populations. And we repeated this for Tuscany and for Rome. And then we simply looked at the phylogenetic tree and just tried to understand first how lizards are related to one another and what these neutral relationships are, genetically speaking. And what we find when we do this by using a genome-wide data set is basically isolation by distance. So we find, again, what we expect, that the Southern Alps individuals are more distantly related to all these Italian individuals. And we see that they do not cluster by syndrome or not syndrome. So it's not the case that all lizards with these green phenotypes are more closely related, but they are more closely related by geography, basically. But then when we select not all loci across the genome, but if we only select the loci that are associated with these syndrome traits, then we find that they do cluster by syndrome. So all green lizards are forming a monophyletic group. And we can also conclude then that the origin of this syndrome really lies close to today's Rome. So what we conclude from this is that the origin must have lying somewhere here, perhaps maybe on an island outside of Rome. And then from there it has spread across the coast of Tuscany, across the coast of Liguria. And we think that it's currently ongoing and that the phenotypes are making their way towards France at the moment. So it's an ongoing introgressive spread of these phenotype that we see here. So now I presented you sort of a snapshot of the distribution and of the background of these phenotypes. And in the remainder of the talk now, I will address 2 questions. The first one is what it is that is driving the spread of the syndrome. And the second one is about the genetic basis of the syndrome. To address the first question, we have done a number of experiments that use these outdoor enclosures as experimental contact zones. So here on this picture, you see one of these outdoor enclosures that's 10 by 10 meters, and that has these pellets and these bruise blocks. And we had 10 of these outer enclosures that we used to experimentally assess how lizards are behaving towards one another. And what we have done is that in 2 consecutive field seasons, we have populated these outer enclosures with lizards of known specific phenotypes. So either we have taken half green or half brown or mixed them in certain other combinations. And then we have done behavioral observations on them. So each lizard was individually marked. We had a number of students positioned around the enclosures that took behavioral observations, and then they reconstructed social networks. So we knew exactly who was hanging out with whom and who was mating with whom. Then at the end of the spring season, after the mating season was over, we collected all animals again and brought them to the lab where the females laid their eggs. And then on these eggs, we could score paternity to know who has sired their offspring. And we also scored hatching success to see how many eggs we had that failed to hatch, basically. And then we complemented this by also running mate choice experiments in the lab to really get the full picture of the intraspecific behavior of these lizards. And I make a very long story very short here because the results were quite unambiguous. So what we find is that when we only have Italian males in outer enclosures with females, then we find that the expression of the syndrome traits is always associated with reproductive success. So the higher a male scores on blackness, lateral blue spots, or greenness, for example, the more offspring they would sire. Second, we find that in these experimental contact zones, green males are dominant over brown males. So here there's dominance is plotted against body size as SVL here. And you see that in green lizards, there's a very strong positive relationship, meaning that they are very dominant over all brown males. And that is true even for very small green males. So even green males that are very small are dominant over brown males. And this higher dominance then also translates to more courtship success with the females here. So that's shown in this plot. And third, when we then score paternity on the eggs that were laid by the females, we find that the mating is assortative, but also asymmetric. And that's shown on these plots here. So very obviously, almost all green females have only offspring with green fathers. Only very few have offspring with brown fathers. And in brown females, they have the majority of their eggs are sired by brown fathers, but quite a high proportion, almost 40%, have offspring with green fathers. So overall, green males are much more successful in siring offspring with the females, not just with their own green females, but they also have high success with other brown females. So what that means is that sexual selection is mediated by male-male competition, and there's male mate choice. We have very little evidence for female mate choice. It's probably everything is driven by behavior of the males. And this also results in some predictions that we can actually test in the hybrid zone, because this makes prediction over asymmetric gene flow in the hybrid zone. And I'm not going to talk about the details here, but we have investigated that, and that's described in this paper. So all the predictions we have had, they actually hold up quite nicely. Okay, so now we know what's driving the phenotypes, the phenotypic spread. But now the question is, well, how, genomically speaking, how we turn a brown lizard into a green lizard. What are the genomic changes that need to be in place to make this phenotypic change? And what has helped us here is that the introgression into this distantly related lineage is basically a natural outcrossing experiment, because we get the loci that are causal for the phenotypes introgressing into a different genetic background, and that gives us a very high power to actually identify those causal loci. So that was helping us in this case here. So the experimental design that we have used to address the genomic basis here is very similar to the design I showed you before already. So we again have one contrast of pure green and pure brown populations in the Southern Alps lineage. So this is the introgression that we think has the highest power to find this causal loci. And then we had two contrasts in the Italian lineage, one in Tuscany and one around Rome here. So again, we had in total, then we had 3 contrasts between pure brown and pure green lizards. And for these, we had a data set of 60 individuals in total, so 10 of each of these sites. And of these, we conducted FST outlier scans and intersected between the different regions to try and find causal loci. The results I show you here as so-called Manhattan plot. So if you're not familiar with them, so what they show is on the x-axis, we have the chromosomes. So it's 18 autosomes and a set sex chromosome. And for each, and along each chromosome, we have plotted basically the strength of genetic differentiation between green and brown lizards, and the higher locus scores on this scale, the more likely it is associated with the phenotype. So we have analyzed the data here separately for the Southern Alps lineage and for the Italian lineage. And just by eyeballing the data, you see that we have a number of loci that are specific for each of these lineages. But we also have a number of loci that are shared between the 2 lineages. And these are the ones that we're really interested in, because we think that it has not, the syndrome has not arisen twice, but only once. So we think that the real causal loci must be shared between the 2 lineages. So we mostly focused our attention then onto this 86 shared outlier loci. And among these 86 genes that are associated with the syndrome in both lineages, we find that a little bit more than half of them had some kind of annotation. So only for these, we could derive some kind of biological meaningful information. And none of these is an obvious coloration gene. I will come back to coloration a little bit more in the end, because coloration is, of course, the most conspicuous trait. So we have focused on that quite a lot. But more than half of these 47 annotated genes have a link to neurocrest cell biology. And that got me very excited, despite that it might not be so obvious to you why, because you might think that coloration is probably caused by changes in pigment cells. Morphology is probably very complicated, but it's basically mainly changes in bone growth or skeletal elements. And behavior might probably have a hormonal or a neurological basis. So probably not much in common, but what they do have in common is that they develop from the same cell type, and that is the neurocrest cells. So neurocrest cells are vertebrate-specific cells. They are called neurocrest cells because they originate in the early embryo at the crest of the neural tube, when the neural tube is just closing. And from there, they detach from the neural tube and migrate out into the rest of the embryo. And there they give rise to a number of different cell types. And among them are pigment cells, fat cells, cells that make the cartilage of mostly the front parts of the skull, and also adrenal cells that are probably related to aggressive behavior. So the fact that, or you can probably appreciate when you see this scheme here, that if you change something in the early regulation of neurocrest cells, that these can have knock-on effects on all these cell types together. So the shared developmental origin in neurocrest cells is probably important for the co-variability of these different traits that develop from this cell type. And this co-variability was suggested by Adam Wilkins to perhaps also explain why some of our domesticated mammals share some certain domesticated features. So in case you have heard of this before, this is basically a similar idea here, but in lizards in our case. So to give you a bit more of a flavor of what these 28 genes are, I just picked a few here, which I think make the most sense. So for example, we have one gene here that is regulating the switch from stem cell state to differentiation in neurocrest cells. There's one gene that regulates neurocrest cell proliferation, so the number of neurocrest cells that you basically have. And there's one gene that is a known regulator for migration of neurocrest cells. So it's all these genes have something to do with how neurocrest cells behave in the early embryo. And we think that tweaking these genes has an effect on all of these traits together. And we're now investigating by using basically developmental biology to try and understand how exactly this works. Okay, so these were the FSC outlier scans that I've now explained to you. But we didn't stop our genomic analysis here because FSC outlier scans are rather blunt tools. So probably a lot of false positives can be found by FSC outlier scans. So therefore, we fine-tuned our analysis and complemented it with more analysis and also more data sets. So on the same data set, we've also looked at patterns of genetic diversity, because we had some certain predictions how Tatima D should change between these different contrasts. And we also collected an independent data set that has some other strengths and weaknesses to complement these approaches. So we used a rat-seq approach, meaning that we didn't look at the entire genomes, but only a subsample set of the genome, but for a larger number of individuals. So here we used 900 individuals and conducted a genome-wide association study to try and see if we find the same outlier locus that we also find in the FSC outlier scans. I don't want to talk too much about the details here. I just want to say that when we intersect across all these data sets and all these analyses, what we consistently find is that there's a single region on chromosome 12 that is always identified as important in all these analyses. And this region is interesting because it was not just identified by our analysis, but it was independently identified by 2 other studies that looked at also neurocrest-derived traits, but in birds. So there's one study that looked at mostly coloration in these parrots, and there's one study that looked at coloration and beak morphology in these redpolls. And they also found that exactly this genomic region is probably the candidate that explains these phenotypic changes. So potentially this region is really interesting, not just from a lizard perspective. And I'll talk you a little bit more through this region because we think that is really, for many reasons, it's quite interesting. So first we find not just a single peak of association, but we find actually 2 peaks, and each of them is quite narrowly defined. They lie on 2 different genes. One is called RAB18 and the other one called ACBD5. It's a little bit difficult to say what exactly these genes do, especially for our lizards, because they don't have a clear description, but it's clear that they have something to do, RAB18 has something to do with cell migration. So it could regulate neurocrest cells, but it's a little bit unclear from the literature how strong that link is. And ACBD5 and also RAB18 are important for intracellular lipid vesicles, and pigments in lizards are deposited in lipid vesicles. So it could be a link to coloration, but currently we can't clearly say if this is the case or not. So we're currently working on that. But both genes are interesting. And since they have this very tight association with the phenotypes, we're looking into that in more detail now. But what is also interesting is the region in between these 2 genes here, because this region, although it's not strictly speaking associated with the phenotypes, it is still quite conspicuous because we have duplicates of 2 genes here. So patched and PKS have 5 and 6 copies in this region. And this, I must say, this is based on the reference genome, and the reference genome is derived from an individual from Spain. So we don't exactly know what our Italian lizards have or have not in this region, but at least the Spanish reference genome has this very high copy number variation here. And just briefly, I show you gene trees of these duplicated genes here, and they just show that they are a mixture of both old and new tandem duplications. So some are as old as reptiles, and some are probably new and probably restricted to that species only, so quite new. So I said we don't really know what our Italian lizards have in that region. But what I can show you is that we have good reasons to believe that things are weird in this region. So when I show you here the cumulative reads depth, so that is just all reads across individuals just piled up one above the other, then for normal genomic regions that just behave like not duplicated just as they should, then they should fall on this 1x red line here. But the region in between those two peaks, so this duplicate region, shows a high number of copy number variation, where we have more read depth than we would expect, and also some regions that are probably absent. So this piece here, present in the Spanish lizard, probably doesn't exist in our Italian lizards. And in order to understand a bit better what is going on in our Italian lizards, we have tried to, sorry, I skipped this. We have tried to solve this question by developing new genomic resources. So we picked one brown individual and one green individual and reconstructed new reference genomes for them, because then we thought we can finally see what is going on in this interpeak region. We got genomes of really high quality. So they were chromosome level, exactly the same, basically the same quality as the Spanish reference genome that was published previously. So all QC metrics told us that these genomes are really great, but our candidate region was not assembled. So here I show you the alignment of the Spanish reference genome with here RAP18 and ACBD5, and here's this complicated region in between. And exactly these parts here are missing in our new genome assembly. So one derived from a green individual and another one from a brown individual. So both of them seem to lack parts here. And why do I know that these parts are missing from the assembly and not missing from the individuals, like really? That is because we have sequence-independent data from the very same individuals. And these are optical maps. So optical maps are a way to study structural variations without sequencing genomes. So but what you get from these kinds of data is basically a barcode of the genome where you can compare different corresponding loci with one another. And when we align those optical maps, so we did this for both individuals, so for the green and for the brown individual, we got two optical maps for this specific genomic region that are aligned here to this Spanish reference genome map that's shown in green in the middle here. And how you read this is basically to look at these gray lines that link different loci from one map to the reference genome. And gray line means basically there's a corresponding locus. And what you see that in orange here are the RAP18 and ACBD5 genomic regions. There's alignment here. So we see that there's corresponding loci between the Spanish reference genome and our optical maps. But in between, so this interpeak region where we have these duplicates of PKS and patched genes, there's no correspondence, but there is DNA. So because the actual length of these optical maps can be directly compared to the maps here. So there's sequence here, but the sequence doesn't align. And we think that the reason why it doesn't align is because it's probably highly derived. There's a lot of small scale duplications and inversions and deletions, and therefore the optical maps can't resolve it. And therefore our new reference genomes assemblies had problems here and simply didn't assemble these regions. So we need to dig a little bit deeper here and probably collect more data to resolve this region for good. But we know that something interesting, I think, is going on here, and therefore we think it's worth to invest some more into this topic. Okay, if I have 4 more minutes, then I can talk a little bit about coloration. So since coloration is, so at least to me, it's the most striking of the traits and one of the more interesting ones that's different between these lizards. And we also think functionally it must be quite important for the lizards. So therefore we investigated the mechanistic basis in some detail. So first of all, when you macroscopically, when you just look at the skin, then you can see that in these green lizards, the scales are actually different. So the scales are either pure green or pure black, while in the brown lizards, you can see that every scale can have a different gradient from different shades of brown, basically. So something here is different in the regulation of how the scales are forming and are colored. And we have not just trusted our own eyes, but we can also measure that by a spectrophotometer measurements. There you can clearly see that the reflectance curves for brown individuals are very different from brown ones. But that's just to say that it's objectively, they are different. It's not just your eyes that trick you. And how color is produced in lizard skin is by different layers of pigment cells. So in lizard skin and also in fish skin, by the way, the top layer is a cell type called xanthophores. So xanthophores are pigment cells that contain carotenoids and pteridine pigments. They are mostly yellow, orange, red in coloration. Under the xanthophores is a layer of iridophores. Iridophores make structural coloration, so they're mostly small, like mirrors basically, that reflect light. And the deepest layer is made of melanophores, which have just black melanin pigments basically. And by using transmission electron microscopy in different colored skin biopsies, so either pure brown, pure green, or pure black, we can try and understand what has to change in this configuration in order to produce brown, green, or black. The result is that, or the answer is that in green skin rather than brown, the top layer has a different kind of vesicle. So in green skin, there's a very high concentration of carotenoid vesicles, which you see have a different appearance in the transmission electron microscopy compared to in brown. These vesicles are almost absent. And in black skin, well, not very surprising, you almost have only melanophores, and the other 2 layers are almost absent. So it's a quite simple explanation for how these colors are produced. So then we try to dig a little bit deeper into these carotenoid vesicles and what they actually are. So for that purpose, we extracted pigments from the skin of lizards, from brown and green specifically. And then we chemically tested which of the carotenoids is responsible for the yellow pigmentation. So the yellow is here, you see in the pigment extract quite bright and shining, so it's very obvious. In the brown, there's almost nothing here. And when we did this chemical analysis, we find that it's either lutein itself or a lutein isomer. And lutein is one of the most widely used carotenoids across lizards, at least. So it's not very spectacular finding, but it's still nice to know what the pigment is actually that makes the difference here. So a green lizard basically has a lot of carotenoid vesicles containing lutein in its skin, and that's absent from the brown one. So that's the difference basically explained. And to link back to all the genetic data that I presented to you, so we also wanted to see if the transcriptomic differences between different colored skin patches, if that is sort of the same kind of genes that we find also in the genomic analysis when we look for differences between green and brown lizards. And the short answer is that there's almost no overlap between these data sets. So the differentially expressed genes that we find between green and brown and black skins Some genes have been previously described in pigment production or pigment synthesis, but most genes have not been described yet. And also our candidate genes, also RAB18 and ACBD5, are not amongst the highest differentially expressed genes. So we think that RAB18 and ACBD5 are unlikely to be really involved in the production of pigments, but they might have some other more indirect role that they play, perhaps. Okay, so with that, I'd like to wrap up and just briefly sum up what I said. So I presented to you that we're dealing with wall lizards of different phenotypes, and these different phenotypes, they vary continuously across the landscape. But these traits, these syndrome traits, they really go together and form a syndrome. I've shown you that the driving force behind this introgressive spread is sexual selection. Green males simply outcompete brown males, and that's why the phenotypes are spreading currently across the Ligurian coast into France, probably soon. The genomic basis is rather complex with many loci contributing, but there's one single major locus that we think needs to be present in order to turn a lizard green. And I've also shown you that a large proportion of the outlier genes that we identify are regulators of neurocrest cells, but none of them is actually these coloration genes that I talked about in the end. And we think that these structural rearrangements in these interpeak regions, that they likely play a role somehow, and we think that they might maintain an evolutionary labile hotspot of variation, but that remains to be tested, obviously. The next steps that we're going to take here, one step that we are already taking now is to go deeper. We want to understand neurocrest cells, how they actually affect phenotypes. We're doing quite a lot of groundwork at the moment. We started to do a single cell RNA-seq between green and brown lizards to try and see if there's extreme differences between the two phenotypes. So that's ongoing. And another thing I'm quite excited about is to expand comparatively into different species. Because although I've focused today on one single species, we have by now sampled 6 other species where we think that the same phenotypes have evolved, perhaps independently. And we're testing that at the moment. So first we test how consistent the syndrome is in other species. And then we want to test if the genomic basis is parallel or if there's convergent ways of turning lizards green and also all these other traits. Yeah, so with this, I'd like to end, and I just want to announce perhaps opportunities for someone listening. So I'm right now based at Lund University in Sweden, but next year I will move to the Max Planck Institute for Evolutionary Biology in Plön. And I'm actually here right now, sitting here. And so next year I'm probably, I will be announcing some positions for starting with me this new group in Plön. And if someone who listened now to this talk and thinks this is interesting stuff, then please just email me and get in touch. I would be happy to receive emails like that. Okay, so thank you for listening, and I leave you with these acknowledgement slides. Obviously, this was a team effort with many people being involved over the years, and we're also grateful to a lot of funding agencies who have supported the Wall Lizard project over the years. And thank you for listening, and I'm happy to take questions from you.